Hello, uh, this is me, David Matherne, and I won an OUR grant for the fall of 2021, and this video should uh, present what I discovered during my research and allow me to share what y'all's investments have yielded. So my project is about Tritrichomonas foetus and finding the drug resistance in the pathogen. So first off, to tell you a little bit about Tritrichomonas foetus, because it's not something that occurs in small talk. Uh, it's a protist that causes the STD trick in mammals. The strain that we decided to look into is the bovine syndrome, which is Tritrichomonas foetus. So the reason we decided to dig into this one was because the human variant of it has more interest and is looked into way more. So we thought that if we explored the bovine variant, then perhaps we could discover something that was maybe overlooked because not as many people have covered upon it. So Trichomonas foetus has a little anatomy. It has three anterior flagella and it has one posterior flagella. The three flagella in the front have the use of allowing the protist to move through very viscous substances such as blood, which it inhabits. And its morphology is that it's a unicellular protozoan. It's spindle-shaped. It lacks a mitochondria, but it has hydrogenosomes. And these hydrogenosomes have an extremely similar function to mitochondria, except hydrogenosomes allow for hydrogen fermentation to occur. And what the two drugs that we'll be using, metronidazole and tenidazole, what they were designed to do, and what they do most of the time, is they target the hydrogenosomes and prevent them from allowing the cell to participate in protein synthesis, which is an obvious function for life because that's how the cells replicate. And so this protist usually ranges from 10 to 15 micrometers to 15 to 20 micrometers in area, and it contains an undulating membrane. So the reason we even covered this in the first place was because it causes STDs in natural breeding cattle. Artificial insemination does not help. When this infects the bulls, there's no symptoms. But when the female is infected, it causes fetal abortions, and it becomes a real issue. And so it's hard to discover at first, but later, obviously, the dire consequences can be the death of the mother and the child. And when you're a cattle farmer, that's the whole point. So it costs U.S. cattle ranchers $650 million annually due to fetal abortions and infertility. And the only two approved drugs for treating this by the FDA are metronidazole and tenidazole, which are going to be the drugs that we're going to be using to test drug resistance. And so my goal is metronidazole and tenidazole have been used for the last 50 years to treat Trichomonas foetus, but over time, due to evolution and natural selection, the genes which allow these protists to resist against these drugs are the ones that have been surviving and being carried down through generations. And so over time, we believe that the resistance of these protists will increase due to certain genes in their phenotype. And so by using a novel genomics approach, we will be trying to discover which anticodon strands cause this resistance to occur. And so what my steps were is we first started by we grew cells using diamonds media, which has tryptose, different organic proteins and compounds that allow allow the cells to thrive. And we essentially we grew the cells in this mixture, and then we isolated them into wells. We treated them with different concentrations of metronidazole and tenidazole, respectively. And we used two different testing group samples. One of the samples was artificially grown, and the other one was extracted directly from an infected cattle. And so after we tested these, we extracted the DNA from these resistant protists, because the only ones that were living after we exposed them were the resistant ones, because it's supposed to immediately kill them. And so with the parasite's DNA extracted, we were able to shear it and then put it inject it into a plasmid and compare it to its the original strain, the genome of an uninfected version of Tritrichomonas foetus, and we would compare the infected, resistant, still living strain with the uninfected normal strain to see what the differences in the codons were. And so, in a video right after this, I will be showing you parts of the lab where I did my experiments. But with metronidazole, with tenidazole, it ended up being stronger and more effective than metronidazole. The strains that the dosage, even at very low dosages, were just yielding, were killing every protist in the vicinity of the well that it was being treated with. But we were able to work with metronidazole, and we used it better. And so we treated it with 1 microgram per milliliter, and then it was a range from 1 microgram per milliliter to 100 micrograms per milliliter. We had about 2 times 10 to the 5th cells per well, and we counted them each day with a hemocytometer in under a microscope. And so we kept doing this. Whenever we saw that not all the Tritrichomonas foetus was dead from our treatment, we would dump out most of the contents, keep the cells, and then pour new media for them to grow and thrive, and then retreat them with metronidazole. We did this about 5 or 6 times to make sure everything, or we were getting the best strain, and that to decrease, error that would occur when this happened. And so they grew. We plotted some growth curves, which you will see in the presentation that goes along with this video. And the DNA isolation, we're actually in the process of that now. Our project is close to being completed, but research takes time, and with time we'll be able to find the best results. And so the genomic DNA, we isolated it from resistant cells, and we checked the gel for quality, and a nanodrop was used for DNA quantification, and we sheared using nucleases. And then what we still need to do is compare to the library to discover the codons and anticodons. But to go back over the conclusions that we have discovered so far, there should be more to come in the future. Is that tenidazole is more effective than metronidazole in a short period of time, according to our research. And the cells, once treated with metronidazole, were able to be, the DNA was able to be extracted out of them. And then, of course, once we finish cloning, the DNA into E. coli, we'll be able to compare the drug-resistant genes. And so that concludes my research for OUR. I would like to thank, of course, the Office of Undergraduate Research for the time and attention. I would like to thank Dr. Kucknoor, my mentor. I would like to thank her lab assistants who helped me grow the cells and keep watch over them. And thank you for everything.